for taking questions. Now let's move on to the next speaker, Dr. Yang Bai from the Institute of Genetics and Developmental Biology, Chinese Academy of Sciences. He will talk about the interaction between plants and the root microbiome. Please, Dr. Bai.
Thank you. Hello, everyone. I'm really happy to attend this event. Many thanks for the support from Molecular Plant and also from the scientific committee. Sorry, don't know what happened. Many thanks for the support from Molecular Plant and the scientific committee to give me this opportunity to share my work about the interaction between plant and the root microbiome. And so in nature, eukaryotes live with a host-associated microbial community. And we call this microbial community as microbiota when we refer to microbes, or we call them microbiome when we also refer to their genomes. And the interactions between host and microbiota happens everywhere. It's from single-cell algae, from fungi, from insect, plant, and as well as our humans. So similar with the interaction between human and our gut microbiota and the plant root associated with huge amount of microbes. And these microbes are very important for plant growth and health. So studying the interactions between plant and the root microbiota is essential for plant growth. And all land plants grow in soil. So 1 gram of soil contains 10 to the power of 6 to 10 to the power of 9 microbes. And all these microbes associated with plant growth in their entire life cycle. And we are interested to understand how plants interact with root microbiota under the natural soil conditions. And we want to know that how plants regulate root microbiota, and we want to know how root microbiota contribute to plant fitness. I work in the plant root microbiota field for 10 years, and I see the entire transitions of root microbiota work from the descriptive to the functional and the mechanistic studies. I was trained in developmental biology until my PhD. And actually, before I graduate, I asked my PhD supervisor, Professor Martin Huska, so what research topic I should do or promising in future. And he told me that the opportunity should be around us, but may only be seen by very few people. And he asked me to try to look for it by myself. And at that time, very lucky that I heard that Professor Schuster-Leifert in Max Planck Institute his group start to work on root microbiota. At that time, Paul's group just transforming from the binary interaction between pathogen and the plant immune system to the complex interaction between plant and the root microbiota to better reflect nature. So I joined Paul's group in 2011. At that time, so the root microbiota research, the entire system was not well established. And later, so my colleague established the sequencing method to detect what microbe associated with plant root. And I did a systematic cultivation of a Arabidopsis root associated bacterial community. So I established a high-throughput bacterial cultivation and also identification system. So finally, we obtained 64% of root bacterial taxa in Arabidopsis root, and also established the synthetic community system to mimic root microbiota assembly in the laboratory conditions. So after the paper published, I think the plant microbiome scientists can do a lot. And they can do the sequencing work and to generate hypotheses based on correlations and data, and then validate the causalities by using cultivated bacteria in the laboratory condition and to investigate the function of this microbe to plant. And I came back to Beijing in 2016 And then I built my own group in Institute of Genetics and Developmental Biology in China, and also the SAPAMS, joined the SAPAMS. And then during last 6 years, I published 33 papers. And so I started my group without taking a postdoc project at that time. So the root microbiome research just start to move from the descriptive work to the functional studies. And so I have technologies, so I know how to do sequencing, and I know how to analyze the data and do the isolate bacteria and do the functional assays. And but what are the main scientific questions? And this is the first thing I was thinking. And the first thing comes to my mind is how to investigate the mechanism between plant microbiota interactions, either from the plant side or from the microbe aspect. So we know that the plant pathogen interactions, like the binary interaction between single microbe and plant, is already very difficult and greatly influenced by environment. And this is nicely explained by Xiu Fang yesterday. And I think we may not want to have more difficulty from the microbio side at the beginning stage. That's why I decided to start with exploring plant genetic pathways that regulate root microbiota, as it may be relatively easy to control. I focus on the interaction of plant and root microbiota using unique plant materials. And today, I will mainly show you results in rice and Arabidopsis. And my work mainly belongs to 3 aspects. The first one is from the microbio side, is the root microbiota's function and association with plant growth. And the second one is from the plant side, is the regulation of root microbiota by plant genetic pathways. And then I also developed a new method in root microbiota research. I start from the first one. So I mainly use rice as a model. And we think rice is a quite unique model to investigate the interaction between plant and root microbiota. So we know that rice is important crops in China and also Asia. And the rice plant mainly grow in paddy soil and may have unique mechanism to interact with root microbes comparing to other land plants. And the genetic resources and tools are available for in rice for molecular studies. And we first examined the dynamic patterns of root bacterial microbiota dynamic patterns and during the entire life cycle of rice under the field conditions. We did a time course experiment, and then we find that from the beginning, the root microbiota of rice changed dramatically and gradually stabilized after 8 to 10 weeks under the field conditions. And this is the time when rice entered the reproductive stage. We also identified the biomarker taxa that's associated with rice growth. And I think this data result provides us a guide when to apply beneficial bacteria in agriculture. And for Asian rice, there are two types. One is called indica, the other one is called japonica. And these 2 types of rice, except they have significant different genomic differences, and they also show many different phenotypes. We are interested in the nitrogen utilization efficiency. And for a long time, people know that indica rice variety show higher nitrogen utilization efficiency than japonica. Like some key gene like NRT121B regulates this process. We are interested want to know that whether root microbiota are involved in nitrogen utilization efficiency of rice. To answer the question, so we grow 68 indica and 27 japonica varieties under the field condition, and we did it in 2 fields as replicate. So we find that the indica and the japonica varieties have distinct root microbiota composition. You will see that they clearly separated, and the result can be nicely reproduced in the replicated field. Comparing with japonica rice, so indica rice variety, the root actually enrich a lot of bacteria related to nitrogen metabolism also enrich the predicted function, which are related to nitrogen cycle. I mean, this data indicates that the nitrogen cycle around the indica root are more active, which are consistent with the indica rice have higher nitrogen utilization efficiency than japonica. Next, we want to identify the genes, rice genes, to regulate nitrogen-related bacterial function. Then we did association analysis between the rice genomic SNPs and the relative abundance of nitrogen-related bacterial or functions. We find that NRT121B show a very strong correlation in both test fields. So collaborated with Professor Chengcai Chu in our institute, we grow the wild type and mutant of NRT121B and as well as the isogenic lines. And we find that these genes really regulate root microbiota assembly. And the NRT121B is a nitrate transporter and sensor, and it helps plant to sense the environmental nitrogen level for rice growth. And to study the function of the root microbes, so we did a systematic isolation. So we isolate root microbes from both indica and japonica representative cultivars. For both cultivar, we can obtain around 70% of root-associated microbes. So using these cultivated microbes, we find that the root microbes can help rice plant to utilize organic nitrogen. So you see that in the germ-free conditions, the rice plant grow badly in the supply of organic nitrogen, comparing with the same amount of inorganic nitrogen. So the indica-enriched bacteria can fully rescue this phenotype, this growth effect. And the japonica enriched bacteria also have effect, but to a less extent. So our result show that the rice plant can coordinate root microbiota to utilize soil nitrogen. And the NRT121B is one of the key genes to regulate this process. We also find that root microbiota is associated with soybean nitrogen fixation and also foxtail millet growth. For the second aspect, so we investigate the regulation of root microbiota by plant genetic pathways. From here, so we are very interested in the plant's metabolite. The reason is the plant use more than 30% of photosynthesized organic carbons to synthesize metabolite in root. And there must be a scientific reason for that. So we ask a question that how do Arabidopsis specialized metabolite shape root microbiota composition? This is a collaborated work with Professor Ann Osborne from John Innes Center. And we find in Arabidopsis genome, there are 5 cluster triterpene pathway. And interestingly, the gene expression of this cluster triterpene pathway are highly expressed in Arabidopsis root, indicating they have function there. And our collaborator identified the entire genetic pathway of this triterpene pathway and also identified the key compound synthesized by this pathway. And we grow the key mutant of the cluster triterpene pathway in the natural soil in Beijing and sequence the root microbiota. We find wild type and mutant plant have significant different root microbiota. You see here comparing to wild type, the triterpene mutant show clearly different root microbiota structure in this PCOA analysis. And the effect is very strong. You can see that the difference, you can see the difference at the bacterial phylum level. So therefore we conclude that the cluster triterpene pathway affect the Arabidopsis root microbiota assembly. And to study, to know which microbes are regulated by cluster triterpene pathways. So we first identify Arabidopsis specific root microbiota members comparing with rice and wheat plant grown in the soil of the same farm. And rice and wheat plant do not have this cluster triterpene pathway genes. And interestingly, we find 52% of Arabidopsis specific root microbiota members are regulated by the cluster triterpene pathways. So either these members are downregulated or upregulated in the triterpene mutant. And this data indicates that the cluster triterpene pathways may be involved in the co-evolution of Arabidopsis and root microbiota. To study whether the triterpenes can directly influence or regulate the bacterial growth, we systematically cultivated root bacteria from aforementioned root samples. Our collaborator purify or synthesize the key triterpene compound. Then we did the binary interaction in vitro. We find that the triterpene can selectively modulate the growth of cultivated root bacteria at the phylum level. So in the tested bacteria, the triterpene can promote the growth of proteobacteria and the firmicutes and inhibits the growth of actinobacteria. And this result are very consistent with our sequencing result, meaning that our data are convincing. And using cultivated bacteria and purified compound, we find that some bacteria can transform some triterpene from one form to another form, or other bacteria can utilize a specific part of the triterpenes as their carbon source. And collaborated with Professor Guodong Wang and Professor Selectively from Stanford University. So we find Arabidopsis derived sister terpene and coumarin modulate root microbiota assembly. And all this data, I think, currently show that the plant secondary metabolites are very important chemicals to mediate the interaction between plant and the root microbiota. So collaborated with Professor Xiaofeng Cao, we find that rice histone methylation pathway regulate hub species of root microbiota. So in rice, there are some bacteria are the hub species in the microbio network. And you can see here they are in the central positions, and they are very important for the assembly of root microbiota. And we find that the STG mutant, which have a defect in the histone methylation, actually in this mutant, the relative abundance of the hub bacterial taxa are severely affected, either upregulated or downregulated. The effect is very strong. So we think this could be due to the broad effect of histone methylation at the genome level. We also developed a new method in root microbiota research. And we always get a request for how to efficiently isolate root bacteria. And last year, Ruben, Paul, and I organized a detailed protocol for how to isolate high-throughput isolate bacteria and identify them or analyze standardized informatic pipelines. And we hope that using this protocol, so people can can efficiently obtain their own bacteria from their own sample during 3 or 4 months. We also established the method to study the absolute abundance of root microbes, microbiota. So now the classical root microbiota sequencing method only generates compositional data based on relative abundance of microbe to microbe. You will see here that, for example, like this microbe has 2 and this 1, 1, 4, and in the end, we get a result like this based on the percentage. But if the relative abundance of this microbe stay, but the total amount double, so we don't detect any differences and compared to this. And to overcome this problem, so we establish a new workflow with spike-in plasmid representing plant tissues. And using our new protocol, you see from these 2 mock samples, actually the bacterial relative abundance are the same, but the total amount increased double actually in the second samples. The classical method do not detect any differences. And if you use our method, so we can precisely detect the increase of all individual bacteria with twofold. And using our sequencing and microbial cultivation technology, we also collaborated with other scientists and to extend root microbiota research to other microbiota research area. And this is a case that I collaborated with Professor Chen Yun in Zhejiang University. And in this case, we work with Fusarium fungal associated microbiome. We try to identify biocontrol bacteria, which can inhibit the growth of a Fusarium pathogen, which is a pathogen for the wheat head blight. And using from the sequencing data, we identify a core Fusarium spore-associated bacterial microbiome. Using the cultivated bacteria, we did a screening against the Fusarium pathogen and identify a Pantoea strains which show the strongest antifungal activity. And the strain can protect wheat growth in the presence of a Fusarium pathogen in both laboratory and as well as field conditions. So we also identify the functional compound responsible for this antifungal activity. It's a chemical called Herbicolin A. And this chemical was reported 40 years ago for antifungal activity. But so our work is the first time to identify the synthesis genes to synthesize this compound. And to make a summary, so my work mainly from this is so first from the root microbiota's function and the description is from microbes to plant, and then the regulation of the root microbiota by plants. This is the plant microbes, and also new method, and also extend our work to other microbiome research fields. And although we did some work, I think the plant root microbiome research field is still an emerging research field. If you can see this clearly, this is a publication from the human microbiome, and this is for plants. So we have still a long way to go. And I think there are mainly three research directions I will put effort in future. The one is to try to find the novel function and the genetic potential of root microbiome in plant in different plant traits. And we want to investigate the plant genetic pathway to keep homeostasis of a plant and the root microbiota. And we want to know the signal and the component mediating plant microbiome interactions. I think the root microbiome work not only review new scientific findings, but also change our way to explore nature. So I think the holistic approach is the best, is the most precise way to describe nature, to see what happens. But this work, actually, if we work in the nature, that's because nature is hard to control, cannot be controlled, so we cannot establish causality between different variables. And that is why we work in the laboratory conditions with reductionist approach to build causalities. And then, however, we don't know that our conclusions stay under natural environment. I think the microbiome technology provides us a unique opportunities to link the laboratory work with the natural environment, at least in the micro aspect. As gut microbiome show diverse function in humans, it may be worth to consider root microbiome in many plant traits. So in the end, I really want to thank our institute and the Key State Library of Plant Genomics and also CPAMS to provide us a special international platform for us to do this basic research. And I'd like to thank all the collaborators and all my group members for their hard work and all the fundings. And thank you for your attention. Thank you.